Buenas tardes a todos. Ya todos tomamos nuestros lugares para retomar las actividades. Tengo un anuncio que compartir. Como última actividad del día, nos vamos a tomar una foto grupal en la escalera cósmica. Entonces, para que lo tengan presente y cuando termine la presentación, no se vayan y nos vemos en la escalera, por favor. Gracias. Le pido al doctor Walter Willett suba al estrado para que nos dicte la conferencia magistral, el impacto de la alimentación en el desarrollo de las enfermedades crónicas, 50 años del Nurses Health Study. Gracias. This is the one going down. No, going down. Yes. ¿Sí pueden poner la presentación del doctor? Having trouble up there.
Back from lunch?
I think they are. I see top of the head of someone. ¿Ya se pudo poner? Creo que ahí están. Todavía no vemos nada. No, there we go. Exactly. Yeah. There you go.
Great. Okay. Thank you. I'm going to give a little bit of an overview of about 50 years. And I guess there was a reason that I was given 50 minutes also, that we get one, 1 minute per year. That's pretty fast. But I will, and a lot has happened during that 50 years. And I'm going to give a little bit of an overview of some things we've learned, some of the operations in our nurses' health study and our other cohorts as well. This is just a brief overview of the timeline of the nurses' health study. I think many of you know, whoops, okay. We enrolled about 121,000 women, registered nurses at that time, and we did talk about already reasons for including nurses. It started off as a very simple study to look at oral contraceptives and breast cancer. That was the primary focus. And Frank Spicer at the bottom was the initiating principal investigator. And Bernie Rosner, the second person there, has been our statistician from the very beginning and still is. I mentioned other people's names here who are actively involved as investigators now, but I could really put together probably several hundred people who have contributed to this study. And again, a number of them in this room that have been among those who really did make this work possible. And it involves many different disciplines and people from students to very senior investigators, not only in the United States, but around the world. What makes this study really different, though, is, as shown here, those red triangles are the dietary assessments. So what's really different is that we have many repeated dietary assessments over time. We've collected blood samples that have also been noted. We collected toenail clippings for trace elements. We have urine samples. We have cheek cells. Together, we have from our 3 cohorts DNA from about 175,000 people, I think. But we haven't, of course, analyzed all of that. We've been doing it as nested case control analyses. The Nurses' Health Study is continuing on. We're still following people. But we realized after a few years we should include men as well. So we enrolled 52,000 health professionals. They're not physicians. They were already in a randomized trial. So these are mostly dentists, veterinarians. And then a lot of the evidence, a lot of the epidemiology was pointing to earlier life risk factors as being particularly important for breast cancer. So we added the Nurses' Health Study 2, where they were 25 to 42 years of age, to really look at younger part of adult life. And then we've also asked their mothers to describe their pregnancy with our participants. So we have details of the woman's, their mother's pregnancy during in the early, so we capture some of the early life experiences of the Nurses' Health Study too, that way, including what if they were breastfed, formula fed, how long, and some simple measures of smoking, physical activity, and some of the socioeconomic variables. And we've also, from Nurses' Health Study 2, when the participants were fairly young, asked them about their diet during their adolescent years, their high school years. And that we've done some validation studies, and it's actually pretty good data. So we are piecing together the full life course that way. And we're also enrolling Nurses' Health Study 2 with one objective being able to get more culture, ethnic heritage diversity, and also aiming for younger women as well. So we have about 70,000 participants in Nurses' Health Study 2, plus 25,000 offspring of Nurses' Health Study 2 that we enrolled when they were 10 to 14 years of age. So we're trying to get at that earlier life period, which does look like it's so important and for which we have relatively limited data. If you'll excuse me, I'm going to weave a little bit of my own personal background into this. Partly it's intertwined fairly closely with the Nurses' Health Study, and especially the dietary component of the Nurses' Health Study. And also, just for those of you who are still in training and younger, we're all in training still, but still if you're younger and in training, some of the things that I did early on that really had some major influences on what I'm doing today. My life would have been quite different otherwise. As you can see down at the bottom left, that's Michigan, the state of Michigan in the Midwest. That's where I was born. And you already heard I went to medical school there. And that's where I really started off. Our families were dairy farmers. That's our grandchild there. Now he's actually 15 years of age. But this is the Willett family farm. It's still in the family, actually. I don't milk the cows anymore. But in fact, these small farms don't exist anymore. Now, my grandfather had 8 cows. There are now like almost 8,000 cows on a farm. It's very different in the Midwest now, not necessarily for the better. And I actually grew vegetables. I'm planting corn here. I did manage to pay my tuition in college with selling vegetables. So, but tuition was pretty cheap those days. I didn't have to sell a lot. One of the early experiences in medical school that I did was with another medical student, a mini health and nutrition survey in an indigenous population in the upper part of Michigan, the upper peninsula of Michigan. And that was really a really important learning experience for me. But I did get started off, this is my first paper, and a bad start academically. I misspelled my name and I didn't catch it. It has 2 T's on the end. And I could never find this paper till a student found it. It wasn't in PubMed. So anyway, be careful about your name when you start publishing. But this was a survey of, this is an Indian tribe that had been pushed out of their native place where they live really 4 times up to land that was very difficult to cultivate, really hard to live in the northern part of Michigan. And I learned one thing was, first of all, we could collect dietary data with a very simple structured questionnaire. And that really got me interested in dietary assessment and realized we could collect data efficiently. And there was a lot of variation between people. Also, even though the numbers were very strong, obesity was really high in this group. And diabetes was also high there, 20%. It was really small numbers, but the actual background in the white population of the United States at that time was only 1.5%. That's like 10 times higher now. But this got me wondering, what's going on here? Why is this population having so much trouble with obesity and diabetes? This was just starting to be in one other group, the Pima Indians in Arizona. There was evidence that there was a high rate of obesity and diabetes there, but this was just the start of the tip of the iceberg of what was going to be really a national and now global epidemic of obesity and diabetes. Another summer I spent here in Tlaxcala, in San Felipe Iztacuistla, and I worked with it was a working experience. We were helping build a school there, but that I learned a lot there too about traditional diet. And this was also, I was able to see the epidemiologic transition. At this time, this was 1971. Pope was crystal clear every morning there. And people were living pretty much like they had lived for probably maybe 1,000 or 2,000 years, mainly basically subsistence farming on maize and corn primarily, with quite a few vegetables there. But almost every week a child was dying in this little village of diarrhea or pneumonia or something like that. But people were active and the adults were probably pretty healthy. We didn't do any formal survey there in nutrition. This was a family I got to know. This was the father here and the grandfather here. They're harvesting and drying out their beans, and then have to sort through them all. And again, this is probably what people were doing for a very long time, up until again early 1970s. And I did go back once. Still, it was actually figuring out it's probably almost 30 years ago. And just in that time, the things had transitioned. Volkswagen had built a plant in Puebla, and the men had gone to work there and had some income, so people were better off. They did have some electricity. They didn't really have electricity or running water here when I was there in 1970. But already people were getting obese visibly. You didn't, again, need a survey. And the people I met were developing diabetes already. So just in that really less than 2-decade period, there had been a major transition. And I did also really learn that for me, it was such an experience. People had such different lives, but they were really same people, like my family back home, which really was, I think, made me connect with people in a different way from around the world. And again, yeah, those beautiful mountains is right in the background there. And this is every morning the wife would bring her maize, that's the molino there, to get them ground up and have their meals pretty much as they would have for long in the past. And by the way, you need a little pulque at the end of the day as well. So that was one very helpful experience for me. And another experience was another elective I had in medical school was to spend a few months in Dar es Salaam, Tanzania. And I really liked it there. It was very progressive. It was an African socialist country at that time. And so when I finished my medical training, I went back for 3 years to join the faculty at the University of Dar es Salaam. And that's my wife, and that's our first child there, who's actually 50 now. And you can see that's Kilimanjaro in the background. It had a lot of white on top. It's almost gone due to climate change, and I'm the one with the white on top now. So that's different. And again, I tried out our standardized, what was a simple food frequency questionnaire in these village surveys there. And you could gain a lot of information, I realized, that way with structured dietary questionnaires. And unlike what people were saying, everybody is eating the same. No, they weren't. There were some very big differences. Little differences in economics made a big change in diet there. We actually did a randomized trial here showing you could improve growth rates of children by treating their ascariasis. But in 1977, I came back and started working on a nurse's health study. And I'm going to describe some of the findings, but also on the other side here, methods that when we first started collecting dietary data, as you know, you don't have any results for a few years in any prospective results. So, well, rather than sit around, I was doing methodologic work or thinking about what some methodologic issues in measuring and analyzing dietary data. 1980, this is our first dietary questionnaire. This is one section. It was only about 81 items long at that time that people checked off these boxes here describing their average intake over the past year. And we found pretty quickly that it was very costly to enter all of that data, and it was also prone to errors. We had some big data quality error problems, so we had to go back and re-enter a lot of data. So quickly realized we needed another method to collect dietary data. So we pilot tested it and switched over to using optical scanning, and that was a big step forward to cut our data entry costs by about 90% and reduced error rate. And we could then afford to make the questionnaire longer, about 130 items here by 1984. So the basic question that everybody asks, well, in fact, I usually say, oh, I can't even remember what I ate for lunch yesterday. But of course, that's not what we want to know. We want to know what was your usual intake over a longer period of time. And we use, like most people, we're using a 1-year time frame. What was your average intake over the previous year? So we did really the first rigorous evaluation of our dietary questionnaire. I won't go into that. It was published in American Journal of Epidemiology, and it actually was republished just a year or two ago as one of the top papers in the last 50 years, because it really did show that we could measure diet, not perfectly, and we knew we could never measure it perfectly, but you could gather, it had a good degree of validity, even with that simple questionnaire. Everybody, as a comparison, 173 people were selected, filled out a weighed diet record. So they weighed everything they ate or measured it if they were away from home, and wrote it down meal by meal. And that was our comparison method. We had them do that for 4 one-week periods over a year in our validation study. And again, not perfect agreement, but quite strong agreement here if you look at cross-classification of quintiles. So that did really provide us with an evidence base to allow us to move forward in nutritional epidemiology. We could show that, yes, we could measure diet reasonably well, but with certainly some errors. One of our first papers was looking at body mass index and breast cancer. And there we were shocked to see with increasing quintiles of BMI, quintiles index, there was not an increase in risk like we thought we were going to see. There was actually a decreasing risk. As you can see, it went from 1.0 to 0.9 to 0.9 to 0.73 to 0.66. And that was really strange, but that's been reproduced. Everybody finds the same thing if you look at premenopausal women. But what's really important, we came to see as our population got a little bit older, it's really the weight gain after age 18 that's important for breast cancer risk. And then the relationship flips around after menopause. And we still don't understand why that inverse relation exists. I have some ideas, but it remains one of the very big paradoxes in the epidemiology of cancer. Back to our methodologic findings, it occurred to me that failure to adjust for energy intake in epidemiologic analyses was not allowing us to separate the quantity of food, overall quantity of food, from the composition of the food. And those are two very different ways of looking at diet. So Mayor Stamford and I did publish this paper on total energy intake that generated actually 10 years of correspondence in the American Journal of Epidemiology, which was good correspondence. It was the way scholarship should work, not trashing each other on the internet. And in the end, it has become really standard to adjust for total energy intake in epidemiologic analyses, because as individuals, that's what we can really change, is the composition of our diet. If I want to double my vitamin C intake, I can't just eat twice as much food. No, you have to change the composition of the diet. If you eat twice as much food, you can't really do that over the long run, but bad things will happen if you try. So this is the method we suggested. at that time using regression residuals to adjust for total energy intake. But then we've also shown you could use what we call multivariate nutrient density models as well. There's not just a single way to look at that. But that, I think, has been, and actually that is one of our most cited papers. The main funding to allow us to collect the dietary data in the Nurses' Health Study, as I mentioned earlier, was to look at fat intake and breast cancer, because it was strongly believed with very great conviction by people in the nutrition area and cancer prevention area that fat in the diet was the main cause of cancers, breast cancer and other cancers as well. But when I looked at what literature was available, it didn't look like the literature was really supporting that, but there wasn't much literature, and it was clear that we needed to do a cohort study, a prospective study. And again, that's what got us funding. So that was one of our early papers. It did get published in the New England Journal, where we did not see any hint of a positive relationship between energy-adjusted fat intake and breast cancer risk. As you can see there, in both age-adjusted and multivariate models, is with increasing fat intake, increasing quintiles, relative risk is going down slightly, not up. And so we're not supporting conventional wisdom. And this drew huge attacks from a lot of people who did not want to accept that fat was not the main cause of breast cancer. Fat in the diet was not the main cause of breast cancer. And this is important because this was driving that belief was driving dietary guidelines. And that our dietary pyramid was initially avoid all types of fats and oils and eat lots of carbohydrate. That was the main nutritional advice in the 1990s and early 2000s. So one of the criticisms was that, again, you have measurement error. If you just had better measurement, you would have seen a positive association. So Dr. Rosner and I did develop a paper, and then Donna Spiegelman joined us for this follow-up paper, to use statistical methods to take the results of our validation or calibration study among 173 participants, take those results and use those to adjust relative risks relating exposure with outcome. And here, looking at fat intake and breast cancer risk. So we could show in that adjusted analysis, this is a simple method, a conceptual method. It boils down to something so simple. It's basically the correction is just to regress your observed intake on true intake, and you use the residuals from that simple regression analysis in your calibration study to correct the regression coefficient. So very simple. We expanded it to be useful in a multivariate mode, but we could show that even when you took measurement error into account and adjusted the relative risk and adjusted the confidence intervals, there was only a very weak, even the upper bound confidence interval was much, the relative risk was very close to 1.0 and was incompatible with the ecological correlations or the animal studies. So by this time we had done enough methodologic work. I thought it would be useful to put this together in a textbook. So this was the first version of the textbook, Nutritional Epidemiology. It wasn't too hard to do. Well, it was a lot more work than I thought it was going to be, but like it always is. But I could just include every published paper there was on nutritional epidemiology in that version of the textbook. There wasn't much out there. Another variable that we looked at from the very beginning was trans fat. I was concerned on the basis of some of the biochemistry and the fact that you were taking essential fatty acids and twisting the shape of those molecules and doing that on a massive basis in the food supply and creating trans fat. I was concerned that twisting those essential molecules was going to change their function in ways that we couldn't predict. So from the very beginning, we actually set up a laboratory with a 100-meter column to measure trans fat in foods because there was no such database. Frank Sachs in our lab oversaw that laboratory work, and we created a database for trans fatty acids in the foods, collected very detailed data on types and amounts of margarines we used, and when we first looked at trans fat and coronary heart disease, the results were, in fact, very disturbing. We did see a relative risk of 1.8 for the top category of trans fat intake in relation to heart disease. This was rejected by lots of journals in the US. Nobody wanted this paper. The food industry didn't want it. The American Heart Association people did not want it. They thought this was a distraction from saturated fat. They actually were against us in the courts that were trying to remove trans fat, but eventually the findings were reproduced, and we worked with advocacy groups to get trans fat on the label and out of restaurants, and as of 2018, it's been illegal in the United States, but by that time it was basically gone, and it's been gone from most of the world's food systems. Now, there are a few places in Asia where there's still trans fat in the food supply, but not very many places still have that. To get a better look at fat and breast cancer, we also created the Diet and Cancer Pooling Project, and this brought together what were all the cohort studies at that point in time. There were basically 7 to pool our data on fat and breast cancer, and that was very helpful. And David Hunter led that paper, and we basically found that when we pooled the data, this was about 5,000 cases of breast cancer by this time, there was no hint at all of an increase in breast cancer with higher dietary fat. And that was really important because the process there, we really brought together all the investigators. It wasn't just one investigator taking the data, but we worked together to, as we have in the polling project, to include the investigators in the planning and the analysis and the writing paper. So we really developed our own interactions, our own self-critique and consensus there by this kind of work, which I really think is the best way to put data together. And it's great that ESMaestras has joined the latest round of pooling data from the for type 2 diabetes. Also, what was really I realized was important was substitution analyses for macronutrients. That if you're going to increase your intake of, say, saturated fat, you're going to decrease something else, and that something else makes a big difference. It's why when you're designing a randomized feeding study, you spend a lot of time thinking about what is a comparison diet, because that will really determine your results. So we did publish really I think the first paper looking at substitution for macronutrients, and Frank Hu led that analysis in the New England Journal again in relation to coronary heart disease. And there we saw that, yes, it really does make a big difference what the comparison group is, as we sort of expected. But basically, if you exchange trans fat for carbohydrate, there's really no significant impact there. That's at the top there. And if you if you replace polyunsaturated fat with carbohydrate, there's actually an increased risk of coronary heart disease. Polyunsaturates are very good, and the same with monounsaturates. But at the bottom there, if you replace trans fat with any other type of fat, then that's going to be good. So I think this was really important. And again, this has become now quite routine, especially for macronutrients, to look at substitutions if you're looking at a macronutrient. Also, the work by David Jenkins and on glycemic index looked to me to be quite important, but had not been incorporated into epidemiologic studies, and it was actually being, I think, misused in ways that didn't make sense. So I thought that looking at developing the idea of glycemic load, which is basically the glycemic index times the amount of carbohydrate, could be more important for some analyses than just glycemic index alone. And so actually, this is Jorge Salmerón showed up at our doorstep and took on this analysis for us. And that was really good. This was, again, what I think is one of our most important papers. And I still do show the slide from this, which I think this really does tell the story. We saw it in all 3 of our cohorts, that with glycemic load going from high to low, that's the from left to right, there is related to lower risk, lower glycemic load being related to lower risk of type 2 diabetes. And on the other axis, I find that thinking of this as a two-dimensional situation. You can make it into more dimensions than two, but this is really the core, that higher cereal fiber intake related to lower risk of type 2 diabetes. And you really don't want to be in that group in the back, which is high glycemic load and low cereal fiber. And the dietary guidelines were pushing people into that category, actually, which did not seem to be a good way to go. And both of these have been shown in other large studies and pooled analyses to be important. But it is important to remember, this is cereal fiber mostly from wheat. And what about maize? Is that going to be the same? So I'm waiting for Jorge or somebody working with him in ESMaestras to update this in Mexico to see, will we see the same picture when we look at maize? It will be amazing, but I don't know what exactly it will show. And we might hypothesize it will be the same, but I'm not totally sure. Sim and Luke did this analysis while working with us as a student. And this is also, I think, really describing what's going on around the world, because a lot of people have said, well, look at China. They said this 20 years ago, not today. Look at China. They're eating all this white rice and they're not overweight, they're not having diabetes, but they were very lean and very active. And had very low BMI. The average BMI in China was 22 at that time. And so if you have low BMI and you're physically or have a lot of physical activity, you're going to have a lot of insulin resistance. But if your BMI starts to go up, you're going to develop a lot of insulin resistance. If you then, on top of that, have a high glycemic load diet, you get very clearly demonstrated metabolic abnormalities. Your glucose goes up, your insulin goes up, your triglycerides go up, your HDL goes down, and you were really entering this sort of pre-diabetic state, dyslipidemia, or you might basically the characteristics of pre-diabetes. And so that's what's happening around the world. We had a few nurses, this is all from Nurses Health Study, who were BMI less than 23, not very many anymore. But most of our population has moved up into that higher category now, or at least close to a BMI of 30 or higher. In the US, the average BMI is about 30, and I think that's close to what it is in Mexico now too. So most of the world has shifted toward that higher BMI. And the point is that even if they kept the same diet, then you could still have an explosion of diabetes there, even if diet doesn't change. But unfortunately, the diet has usually become more refined. So time for another update of nutritional epidemiology. So I thought that's just a few additional references, but that wasn't so easy because there were a lot of publications, so I had to go through paragraph by paragraph to take out some old things and add new things, so it ended up with being a lot more work than I thought it was going to be. A lot of our work in recent years has been on dietary patterns, and that does seem to be a useful way of looking at foods. But let's see, this is looking at our second validation study that we included in the second edition of the textbook here. And we had been able to expand our questionnaire, as I mentioned, and this was validating our more expanded questionnaire. And then we could look at the average of multiple dietary assessments. If you look at the last column, the 1980, 1984, 1986 food frequency questionnaire average, and compare that with the average of diet records over 6 years. And there, when you statistically account for variability in the diet records, the comparison methods, the correlations now are really very good, 0.83 for total fat and 0.95 for saturated fat. So by doing the two things, by improving the questionnaire, but also then averaging multiple questionnaires, you can really get quite high validity that way. And so it really does, again, make the point that having repeated questionnaires is very useful. We also did some analyses looking at the population attributable risk or population attributable fraction for coronary heart disease. I mentioned that earlier. Today, Mayor Stamford led that analysis, and we could see that by not smoking, keeping BMI below 25, moderate exercise, a good dietary pattern, and avoiding heavy alcohol. Actually, the little alcohol was related to a somewhat lower risk. It looked like, well, first of all, only 3.1% of our population fell into that low risk group, but we could estimate if everybody adopted that pattern or close to it, that would prevent about 82% of coronary heart disease. Again, making the point that this is really a preventable disease by diet and lifestyle factors. And we did not include using pharmacologic methods in this analysis. We looked at that in some other analyses. We've also looked at overall diet quality in our cohort studies, and this is in relation to a composite of major chronic disease. And we used this approach to assess the validity of the dietary guidelines and the dietary pyramid that came out in 1993. And it looked like to us this was not giving people the right advice. Again, the emphasis was avoid all fats, load up on carbohydrates. And so we actually scored people's participants' diets by adherence to that national dietary guidelines that we thought were off target. And that was useful because we could show that after you adjust for smoking and physical activity up at the top there, then there's really no relationship between adhering to the dietary guidelines and risk of major chronic disease. So this really sort of showed in an objective way that the guidelines were not valid. You would hope the guidelines, if they were valid and people adhered to them, that their risk would be lower. So that was helpful to show it, and it did put pressure on the dietary guidelines process. I also, we again talked about communication. We had enough data by this point in time to share this more with the general public. So I did that in terms of a book for the general public called Eat, Drink, and Be Healthy. That's the book there. This was really the first time somebody in mainstream nutrition questioned the sort of the fat hypothesis, and it was radical at this point in time. Got again a lot of negative feedback, but actually that's what the most recent dietary guidelines committees have come to the same conclusion, that it's really emphasizing the quality of the carbohydrate, the quality of the fat, and now even more important, the source of the protein as well. So we did a similar analysis like for coronary heart disease to look at the population attributable fraction for type 2 diabetes. And this was Frank Krug led this analysis. And what we saw, similar combination of risk factors there, that we could prevent about 92% of type 2 diabetes by diet and lifestyle. And these are not extreme changes. These are things that people were making in real life and could have major impact. And again, just the fact that people didn't have diabetes or the rates were very low just 50 years ago is obvious that this is not an inevitable disease, but this we are showing in real populations what you can do to bring your risk down to very low. We've also looked, given that the dietary guidelines were off target, I thought we should provide some alternative to the national dietary guidelines. So Margie McCullough, who was a great student with us, dietitian, and then went on to American Cancer Society, helped us put together the alternative healthy eating index, which we've used and updated a little bit since that time. And we were able to show there that adherence to that was related to better health outcomes, particularly for cardiovascular disease. There, as you can see, there's a substantially lower risk with better adherence to the alternative healthy eating index. But for cancer, there wasn't, it was pretty null. And it really, I think that's persisted, that at least in the relatively short term, the effect of diet on cancer overall is not as strong as it is for cardiovascular disease. But I'll come back at the duration and more nuanced look at diet is important. Also, we've looked at soda consumption, sugar-sweetened beverage consumption. This is quite an important paper, I thought, done by one of our German students, Matthias Schultz, looking at sugar-sweetened beverages and risk of type 2 diabetes. And there we could see with one or more servings per day, relative risk of 1.85, which that's a pretty substantial increase. And the gold bar there is after adjustment for BMI, but BMI is a mediating factor. So what this is suggesting is there's a metabolic impact of sugar-sweetened beverages above and beyond the weight gain. And clearly, in other analyses, sugar-sweetened beverages are the single most important factor contributing to increase in body mass index. So thinking about all the interventions we might do, I think Mexico got it right of putting the tax on soda. That is the big target. That's not the only target, but it is the biggest and most important and actionable target here. Oops, I'm sorry, I went backwards there. Wrong direction. There was a belief really pushed by some colleagues at the National Cancer Institute with case-controlled studies. If we just ate 5 servings a day of fruits and vegetables, that would drop cancer rates by 50%. That was almost too good to be true. And when we looked, it really wasn't true. It was based 100% on case-controlled studies at that time. And there is a serious risk of bias there. We could see for cancer, it was basically flat for total cancer incidence. And it means that, again, we have to look at other ways to reduce cancer. As it turns out, the biggest single nutrition factor is overweight and BMI for cancer incidence. But for cardiovascular disease, there really was a pretty clear benefit with higher fruit and vegetable consumption. Then we went on to look at substitution for, oops, oh, darn, sorry, pushing the wrong button here. Substitution analyses for foods, major protein sources. After doing substitution analyses for macronutrients, it occurred to me we should also be doing that for foods that are carrying a considerable amount of energy and for red meat. And so if you're not going to eat red meat, you do make a conscious choice about what am I going to eat instead? Is it going to be fish or poultry or beans or nuts? And so when we did this substitution analysis, we could see quite clearly that there was pretty much anything replacing red meat was beneficial. But if we didn't do substitution analyses, then by default, the comparison is other foods in the diet. And that mix in our populations is not very healthy. It's a lot of refined starch, unhealthy fats and sugar. So red meat is not too much different than those unhealthy calories. It's really important to do these substitution analyses. You can see the confidence interval for beans is very wide there. We just don't have enough bean consumption. And that's why we're really waiting for ESMaestras to come out and do this analysis. That's going to be very helpful. Also, as we've gone into these analyses, the importance of time has become more apparent. And in some ways, that we were so naive. We wrote a grant in 1980 to get funding to look at diet and breast cancer. For NIH, you're supposed to have the answers in 5 years. But at 50 years, we're still just really understanding some of these diet and cancer relationships. You really do need this long view. Here for folic acid and coronary heart and then colon cancer, for example, we had to go out to 12 to 16 years to see that there was an inverse relationship with folic acid. And the same pattern, although with a shorter latency, we see for adenomas as well. So we're routinely using those, and without looking at specific latencies, you could miss important benefits or adverse effects of aspects of diet or smoking. Actually, Ed Giovannucci showed very clearly that smoking 30 or 40 years before the diagnosis of colorectal cancer. It's not when you smoke 10 years before the diagnosis of colorectal cancer. That's irrelevant. Well, the time came to update the textbook, and our son became a photographer, so I had him take pictures about food, and this is our extended family. That's our cute little grandson down in the left-hand corner there, but he's 15 now, so I'm time to update the textbook again, and I just can't get enough energy to do it. Whoops. But what I'm going to do, I'm actually supposedly on sabbatical now, and I'm going to do sort of update annotated readings for each chapter and put them online to bring things up to date a little bit, because the fundamentals have really not changed appreciably. We then have gone on to look at diet earlier on, now that we're having some follow-up with diet during adolescent years, and to look at breast cancer. And there we see associations with breast cancer that we just didn't see clearly looking at midlife diet here. And that in general, replacing red meat in the diet again was related to lower risk of breast cancer. And if you put it all together, like at the bottom, replacing a combination of nuts, legumes, poultry, or fish for red meat, there was a pretty clear inverse relationship with breast cancer risk for adolescent diet. And we see that fruits and vegetables during that period are also more important. There's also been, I think many of you are aware of debate going on. I think it's resolved now, especially with some people, statisticians at the CDC, who have been claiming that it's better to be overweight or a little bit obese for total mortality, which just didn't make sense from a mechanistic standpoint. And so we've looked at both weight in relation to mortality, but also how to study diet and weight change. And we don't have time to go into the details here, but the bottom line is it is sort of like to emulate a trial here. You look at change in change in diet in relation to change in weight over time. If you just look cross-sectionally, there can be a huge amount of reverse causation that you need to get around. So we did a paper looking at various aspects of diet in relation to change in weight. And that's what ESMaestras has also done a similar analysis, looking at sugar-sweetened beverages, looking at change versus change, which is the way to do it. Here, fruits and vegetables in general are related inversely to weight change, but some of the foods are related to higher weight gain, and particularly potatoes. The three colors are the three different cohorts, and there's quite good consistency here that especially peas and corn were related to higher gains in weight, very different than for most fruits and vegetables. So this does make me a little bit concerned about having a lot of maize in the diet. Now that's mostly sweet corn. It's not the same as tortillas, and what is field corn, not sweet corn, to make those. But it's just all good reason to be able to look at these issues in Mexico. We've also then more recently looked at major protein sources in total mortality. Here, this is Mingyang Song, and what we see here again, replacing different foods, replacing red meat, quite consistent inverse relationship. So again, generalizing the substitution analysis to foods is really important. And interestingly, that was using the updated the updated dietary data. We would have missed a lot without the updated repeated food frequency questionnaires. And we've looked at type of fat in relation to total mortality, saw a very similar pattern to what we saw for incidence of coronary heart disease. That blue line at the top is trans fat, fortunately gone off the table, all in relation here isocalorically to carbohydrate. Saturated fat, weak inverse positive association, but mono unsaturated inverse, and very strong inverse relationship with polyunsaturated fat in the diet. And again, this is just an example. This we didn't publish. What I just showed you is on the left, but if we only use baseline diet, that's what we would see. We would have missed almost all the action there, that we would have missed trans fat entirely. We had would have had MUFA going in the wrong direction, and we would only have seen a weak inverse relationship with polyunsaturated fat in the diet. So again, really important to have the repeated dietary assessments. We did get funding from NCI to look at relative validity for different methods, 24-hour recall, food frequency questionnaire, or 7-day diet record. And a lot of data has come out of this. If you look over at the right, beta-carotene is pretty typical here. Basically, we're looking at which method has the strongest correlation with a biomarker, and that presumably would be the best method. And we were careful about the timing of these methods. Over on the right, 2 weeks of weight diet record, that is the best. But that costs us about $1,000 per week per person to collect and analyze. So it is, I think, the best gold standard. 1 week, the blue line there from the right, is almost as good as 1 week for a lot of nutrients. But then the next is 2 food frequency questionnaires or 1 food frequency questionnaires, just being a little strong correlation. And so basically that is really showing that costs us online almost nothing to collect that and analyze that dietary data versus, $1,000 per person. And that's why the method, it turns out this is a really good method if you're careful about it and careful about how you analyze the data. And then these 2 columns, that's 4 days of diet recall and 1 day of diet recall, much less useful because of the great day-to-day variation in intakes of most nutrients. So how can it be that just this simple sort of primitive methodology, even this paper food frequency questionnaire, how can it be so good? Well, one of our colleagues at Harvard did estimate that the brain includes about 86 billion neurons and 100 trillion connections. And we're basically interrogating that brain. And that's still a pretty powerful organ. It's subject to biases, but that's why we do prospective studies. So it's not biased in relation to disease outcome. And there's no, people are enthusiastic about wearables and visual imaging of plates and stuff like that, but that cannot capture what you ate over the last year. You can get that from our simple questionnaire. Now, maybe some hybrid methods will add a little bit, but basically we are capturing most of the information for most variables. Now, for things like contaminants or some of the processing of foods, unless we were like trans fat, really going after those in details, we may not be able to get. So careful use of the questionnaire and biomarkers is going to be the best approach. There's no one single method that's going to be optimal for most. But in general, the food frequency questionnaire, carefully done, has been still the workhorse of cohort studies, and for good reasons. Just to keep in mind, some of the analyses we're doing now are really going down to specific foods. Now, and I think looking at fruits and vegetables has been really crude. What does a carrot have in common with broccoli or with cauliflower? Really nothing nutritionally. And they are likely to have different biological effects. So we are doing analyses now. This is looking at breast cancer. You need big numbers. So we're running about 10,000 incident cases of breast cancer in that analysis. And it does show that high carotenoid vegetables and cruciferous vegetables were related to lower risks. Now we're also spending quite a bit of time looking at cognitive function. As our cohort gets older, as our population is getting older, there's a lot more interest in helping to maintain cognitive function. And we see, like most studies do, I think there are more than 25 studies now that have looked at Mediterranean diet and cognitive function. In general, we do see inverse relationships. But what is it about the Mediterranean diet? We're digging down and looking at details there. Again, it's good to have that big picture view, but understanding that in detail, something important is happening there, and we'd like to know more about it. So that's just a little brief sampling of things that we've looked at, but we've really had people, many of these analyses done by specialists in the area looking at kidney disease, urology, rheumatology, actually thanks to Mauricio, cardiology, infectious diseases, eye diseases, endocrinology, audiology, otolaryngology, dermatology, primary care, all these fields, we've had impact on practice there with findings from the cohort studies. One methodologic area that we've been looking at more recently is combining measurement error correction with repeated measurements. And until recently, the method that we had with Dr. Rosner was only useful for correcting a single measurement. And of course, we get benefit from repeated measurements. So now we can do both at the same time. And this is just looking at red meat and type 2 diabetes. And by doing this measurement error correction, we actually get quite a big increase in relative risks. I want to say, and just quantitatively there, it looks like for red meat and coronary heart disease, if we have just a single measurement and then long-term follow-up, we're underestimating our relative risk by about 3- or 4-fold. It's not just trivial amounts. So the associations that we see in a lot of meta-analyses that mostly have one measurement of exposure, many of those are very seriously underestimated. I did want to mention just briefly some of the work I've done with the Eat Lancet Commission. And that was, as I think many of you know, brought together to define or identify a pathway to be able to feed what will be about 10 billion people by 2050, a diet that is both healthy and sustainable. And the Eat Commission and Lancet did get funding to let us bring together 35 scientists to look at this issue from different parts of the world and from nutrition, agriculture, earth sciences, sociology, multiple different disciplines. So this was a big work. And Juan Rivera joined us for our latest version that was published in 2025. It was really great to have his input there. So we identified that a dietary pattern that, again, I don't have time to describe in detail, but it's pretty similar to a traditional Mediterranean diet. We've called it plant forward or flexitarian, but allows modest amounts of red meat not more than about once per week though, and dairy with about 1 serving of dairy a day. But again, mostly whole grains, fruits, vegetables, nuts, legumes, soy products. So we could go back to our cohort and score everyone's diet according to adherence to the planetary health diet. And then this is looking at deciles. And we can see here, this is women that with increasing score, there's increasingly lower risk of overall mortality is what we're looking at here. Whoops, Debbie put her note at the bottom. That's the figure I meant, and I didn't change it. So we could see that there would be substantial improvements in overall mortality. And every major cause was lower with better adherence, including neurodegenerative disease, cardiovascular disease, cancer. Actually, the strongest inverse association was with pulmonary disease, interestingly, and even in never smokers, which is quite important. But we also went on then to look at environmental impacts. And so we could score each person according to their contributions of greenhouse gas emissions, fertilizer use, cropland use, irrigation water, and they were all lower, but the cropland use is especially important because, and it's basically very simple, you're feeding legumes and corn to people more than feeding them to animals or making ethanol out of the corn. And that in the US, this is US data, would lead to about a 50% lower land use, which is huge. Basically, you could let us reforest, let us a good bit of land. You could use it for other purposes as well. And people would be healthier at the same time. So then we went on to look at the global picture. What if the whole world shifted to that planetary health diet? How much would be the impact on total mortality there? And so we used dietary data, in this case from the Global Burden of Disease. The second time we did it using some modified FAO data to score every country's diet. As you can see around the Mediterranean diet in Southeast Asia, it looked, that was the best, but even the best countries were far from ideal diet. And in Japan, still way too much white rice, way too much sodium, and not enough fruits and vegetables. So everywhere could improve, but there were big differences among countries there. And Mexico, yeah, about as bad as US. When we updated this actually for 2025, Brazil didn't look quite so good. I'm not sure what the difference was there. One thing we realized is that diet quality itself is, I mean, the dietary data in almost every country could be improved. None of the data are ideal. But we could take the relative risks from our cohort studies and apply them to current national diets and estimate that if we made this shift to a score of 120 or out of a possible 140 on the planetary health diet, that would prevent about 27% of deaths in adults globally, which comes to about 15 million deaths per year. And in the United States, our diet's a little bit below average quality. It would be about 31% of adult deaths could be prevented per year. I didn't look it up for Mexico, but the data are available online by country as well. Actually, it's a supplement to our first paper. One of the things we did and wanted to emphasize in the second report that just came out was to show how this can be put into place. One of the critiques was, oh, you're just talking about Mediterranean diet. Well, we have the best, most complete data from the Mediterranean area, but we wanted to show examples of how this could be put into practice starting with the traditional diets of lots of countries around the world. And we're making a whole collection. I actually have about a dozen pictures like this. This is from Mexico. And obviously, you can use Mexican foods and flavors to put together really delicious, healthy, and sustainable diets. And the same for pretty much every part of the world. North America U.S. was a challenge, though, and we did actually make a beef stew with just a little bit of beef in it there with lots of vegetables that could be healthy and sustainable. One analysis we've done within the Nurses' Health Study that I thought might be interesting here, it goes back to our study in the Indian population. And when I was a medical student, we are mostly have people of European ancestry, but we still have enough Asian, Hispanic, and African American participants to look at with common diseases like type 2 diabetes. And this is a paper we published a while ago. Actually, obviously, Hispanic it doesn't mean much except you speak a certain language. It's the most unsatisfactory, but this is the way the U.S. Census Bureau collects it. And it will be really good to break out that can be broken out by genetics now, although we're not going to have enough power to look at subdivisions of that. But what is interesting is that this is sort of like a future scope in some ways. If people come from different parts of the world and have the same diet as the rest of the European population of the United States, we can control. The thing we can do here is we control very in a detailed way for smoking, BMI, physical activity, diet, everything we can see related to type 2 diabetes. The rate in Hispanic population in our study is about double the European population, and Asian and African American rates are still quite elevated as well. And this is playing out as we see what's happening in the US and around the world as well. So one question was, do the dietary data apply to across the groups? And we had created a diabetes dietary risk reduction score, and we could look at that in our we created that score in our overall population. It's pretty much like the Mediterranean planetary health diet, except it emphasizes nuts and, of course, no sugar sweetened beverages, and emphasizes blueberries and a few other things we've seen related to diabetes risk. But we can see that the same healthy dietary pattern does apply across the population groups as well, which is not surprising. But the good news is that it looks like everybody can be at low risk of type 2 diabetes if we adopt a healthy diet and lifestyle. But obviously, more data from Mexico is going to be really helpful there. And finally, now we're left up. We thought we had been making progress, and now we have the MAHA dietary pyramid, which came out, and it's just a little bit insane with a lot of other things coming out from Washington. And it does promote some fruits and vegetables, but it really emphasizes animal sources of protein, which is not the right way to go. And if you really look hard, you can find a peanut here, but it really is an overall emphasis on a very big increase. It's suggesting in total protein, but animal protein, which is not the way to go for reducing diabetes risk. So that's, again, a quick overview. Just to think about the future, there's still a lot to do. We haven't learned anything. We can see a big picture of where we should be going, but we still have a lot to learn about earlier life exposures and long latency. At 50 years, we're just having enough ER negative breast cancer cases to have a good look at that. The brain, a lot going on there. Hard to study, but we are learning about it. A lot of reverse causation we see. Have to be really careful. And that's very important. We do see, as I mentioned, it looks like there are some important effects of diet and lifestyle. If you look at our publications over the last 5 years, a lot of them have incorporated omics, and I didn't really show any of those, but really diet has to be at the center of that still, and partly for reasons that Rui mentioned, that that's really what we can modify most. And the bigger picture is that if we stratify by genetic risk scores, like a healthy diet's good for everybody, it has more impact on the people who have a higher genetic risk. That's the general pattern we see for a lot of outcomes now. So it does raise a little bit of concern about the focus on personalized nutrition. And actually, that can get in the way of public health, because our main strategies are taxes, restrictions, social desirability, and social norms. And if everything has to be personalized, then those strategies go out the door. So I think we have to be really careful about that. We can learn a lot from the omics, but about mechanisms and some things. But it's still, we really, I think, do need to keep diet at the center of our analyses. Filling gaps, knowledge versus practice. A number of people have commented on that. For sure, we need a lot more research on how do we do practice the best way. And we do need to look more at various socioeconomic groups and geography. Again, big blank spots that Isabel showed for Africa, nothing, and really nothing in South America yet. And climate change. We need to keep our eyes on that, because nothing else counts unless we deal with that. So thank you. So I have time for a question, but no, go over it.
No, creo que tenemos tiempo para un par de preguntas. Si alguien quiere preguntar.
We have a question. I mean, go.
In an evolution from focusing on nutrients, then there was a change focused on foods and then patterns. But maybe for some policies, we need go back to nutrients. That's my impression. I mean, you didn't show much about micronutrients or that possible are substitution, fortification. And I know that you were dealing with that early in your career. So I would like to understand why, if micronutrients still focus on diet and chronic diseases.
Yes, good question. And I think we should be looking at all three all the time. We should be looking at nutrients, foods, and overall dietary patterns. And they each have their advantage. Because as I was sort of implying, even just within the fruit and vegetable group, they're not all the same. And you could easily miss out on some. You could be eating 5 a day and miss out on some that have important benefits. And ultimately, just from a knowledge standpoint, most of this is operating by nutrients or other phytochemicals there. And we may miss some of that with something as sort of crude as the overall dietary pattern, including some things that are related to not food per se, like selenium and iodine and things like that. If you're going to fortify or do supplementation, you really do need to know the things at a nutrient level. And fortification and supplementation have had important roles in nutrition. We shouldn't forget about that. So you're right that more recently there's been a lot more emphasis on the pattern, as we did here. One of the advantages is that gets around confounding a lot by different components of foods. It's around that source of confounding. So, and it also inherently incorporates a substitution, even though not in a formal way. But if you have a pattern that emphasizes a lot of plant protein sources, it means the patterns generally then are lower in animal sources of protein. So it's a lot of this is incorporated into the pattern. So anyway, they all have their advantages and disadvantages, but coming at a complicated topic with different perspective, different lenses, I think gives the biggest insight. It's sort of like the old ancient Indian story of different blind men palpating an elephant. You get a very different understanding of the elephant depending on where you're touching the elephant. And I think that's sort of like nutrition. You have the best understanding coming out from multiple directions.
And what about the updating of the nutrient contents of foods? Because it's also a problem because we are based on some historic data, and we were looking for Mexican data as well. And I don't know how you're dealing with changes in nutrient contents in foods, especially in processed foods. So are you doing something within the nurses to understand the nutrient contents of foods and the changes in processed foods?
Yeah, so we actually update our database every 4 years along with the updated dietary questionnaire as well. So it's not perfect for some things like type of fat. Until recently, we've been actually going to the grocery stores and getting off the shelves the different margarines and different breakfast cereals and cookies and things like that and bringing them back to our lab and analyzing fatty acids. I think we're the only place in the world that's done that. It obviously costs something to do that. And there are fortification practices that have changed a lot over time, folate fortification, for example. So we do incorporate as much as we can in that. And if you're interested in a specific nutrient, you really want to dig down deeply and maybe work with a laboratory. First of all, look at the major sources and maybe work with a laboratory that can measure those and incorporate that into the databases.
Okay. Ya no tenemos más tiempo. Una más y ya.
Very brief. No, thanks, Dr. Willett. It's great to see how the cohort studies that you described has been like hand by hand with the development of nutritional epidemiology. You were very subtle and very polite when referring to the new dietary guidelines, but what is your guess? I mean, now looking at everything with the planetary health lens, what may happen in the future and how your cohort studies may help like to see the effects of these guidelines?
Yeah, it's a big problem. I think the rest of the world is asking that same question, that what's going on there? I sort of, you could maybe say if you believe that vaccines are the major cause of autism, this is the diet for you. It's about that level of credibility. But it's obviously counter to a vast amount of evidence. And I think there are a lot of organizations, the American Heart Association, Diabetes Association have pushed back against this. It's like the states are setting up their own vaccine committees. So I think we have to go around at the moment, and hopefully there'll be some sanity restored at some point in time. But it does do a lot of damage, because this is not just recommendations. This is meant to be the guide about what's served in schools, what's served in all governmental institutions. So it has a direct practice implication. Now, a little bit there does depend on, you could interpret some of these a little differently depending on what you wanted to do. So I think some people are trying to take advantage of some of the potential flexibility here. But yeah, it's just one piece of an extraordinarily problematic situation. Yeah. Thank you.
Muchas gracias, doctor Willett. 50 años de ciencia que han cambiado la forma en que entendemos la salud y que han inspirado a generaciones enteras y muchas por venir. Con esta conferencia concluimos las actividades académicas del día de hoy.